Today, today we'll take up C4 plants. Before going on to understand C4 plants, let us revisit C3 plants. As we have already studied, C3 plants are those plants where the initial product is a 3-phosphoglycerate with 3 carbon atoms. These plants are also known as temperate plants. These plants can be annual, perennial. They are highly rich in proteins. Wheat, oats, rye, they are some of the examples of C3 plants. These plants carry out the C3 cycle to fix carbon from carbon dioxide into 3 carbon sugars, and plants over a period of time transform these 3 carbon compounds into nucleotides, amino acids, and complex sugars. Most of the organic matter is created due to the process of carbon fixation. The sugars produced in the C3 or Calvin cycle is also used up by plants for storage of energy. The C3 cycle is often referred to as a light independent reaction to the process of photosynthesis because the C3 cycle is fueled, it utilizes ATP and NADPH, which are generated during the light dependent reactions of photosynthesis in the chloroplast. It efficiently eliminates greenhouse gas from the atmosphere by fixing carbon dioxide. The C3 cycle collects energy derived from sunlight for long-term storage of sugars that can further be used by plants and consumed by animals, which form the foundation of the food chain. All plants use the photosynthetic carbon reduction cycle or the Calvin-Benson cycle for carbon dioxide fixation. This cycle is operative in the cytoplasm of all mesophyll cells which are present in the leaves. In Calvin-Benson cycle, ribulose 1,5-biphosphate is carboxylated by the enzyme Rubisco. Rubisco catalyzes the first step. It adds a carbon molecule to a 5-carbon compound ribulose 1,5-biphosphate to form 2 molecules of 3-phosphoglycerates. 3-phosphoglycerate is a 3-carbon compound. For this reason, this process is also known as the C3 cycle. Rubisco, which is the first enzyme catalyzing the Calvin cycle, its full form is ribulose 1,5-biphosphate carboxylase oxygenase. This enzyme Rubisco shows a duality. It is a major disadvantage in the C3 cycle because Rubisco can carboxylate as well as oxygenate ribulose biphosphate, the substrate of the Calvin cycle. Instead of carboxylating ribulose biphosphate and leading on to the production of glucose, it oxygenates this substrate ribulose biphosphate. So this is a process which is a waste. It does not lead to any gain in the form of some photosynthetic substrate being synthesized. Environmental variables such as high temperature and drought, they result in the increase in the oxygenase action of Rubisco. Two key factors determine how frequently whether oxygen will be used by Rubisco or carbon dioxide will be chosen by Rubisco. And the first of these factors is the relative concentration of oxygen and carbon dioxide. When stomata open, carbon dioxide diffuses in and oxygen and water vapor diffuse out. Photosynthesis is normal and photorespiration is minimized. However, when a plant closes its stomata, for instance to reduce water loss by evaporation, then oxygen from photosynthesis builds up inside the leaf. Under these conditions, Photorespiration increases due to the higher rate of oxygen as compared to carbon dioxide. Of the two key factors, the second is high temperature. Rubisco has a higher affinity for oxygen when temperatures increase. At mild temperatures, Rubisco's affinity to bind carbon dioxide is about 80 times higher than its affinity for oxygen. However, at higher temperatures, Rubisco is less able to tell the molecules apart and binds oxygen more often. Hot, dry conditions tend to cause more photorespiration unless plants have special features to minimize this problem. Photorespiration, which is the outcome of the oxygenase action of Rubisco, is a wasteful process. Rubisco makes up 30% or more of the soluble protein in a typical plant leaf cell. Therefore, the loss incurred by way of photosynthesis is considerable. Photorespiration wastes energy and decreases sugar synthesis. Photorespiration can result in losses of between 25 and 30% of the carbon fixed. So up till now we have seen that the Calvin cycle is operative in C3 plants, and the enzyme which catalyzes the first step of carboxylation of ribulose biphosphate is Rubisco. However, Rubisco can either go down the Calvin cycle pathway or it can go down the photorespiratory pathway by oxygenating ribulose 1,5 biphosphate. C4 photosynthesis reduces photorespiration by concentrating carbon dioxide around Rubisco. To ensure that Rubisco works in an environment where there is a lot of carbon dioxide and very little oxygen, carbon dioxide leaves, they have differentiated two partially isolated compartments called mesophyll cells and bundle sheath cells, which you can see in the diagram in the second C4 plant figure, where there is one mesophyll cell as well as a bundle sheath cell. They are shown. However, in spite of being separate, they are however connected by means of plasmodesmata. C4 plants include many tropical grasses and are among the world's most important crop species. Examples are maize, sugarcane. There are about 10,000 grass species worldwide, and of these 10,000 grass species, 50% are C4 plants. Although C4 plants are about only 3% of the total flowering plants worldwide, their productivity is very high. C4 grasses in savanna regions, they comprise of about 13% of the Earth's vegetated surface and are responsible for 20% of the global photosynthesis. This is a world map, and on this map, the savanna regions of the world are shown. The dark green patches are the savanna regions, that is the grasslands. Steps in the C4 pathway. To avoid photorespiration and excess water loss, tropical plants operate an alternative form of photosynthetic dark phase in which carbon dioxide is fixed by enzymes other than Rubisco. That is, in the leaf mesophyll cells, enzyme Rubisco is absent, and here carbon dioxide is fixed by a different enzyme, which is PEP carboxylase, phosphoenol pyruvate carboxylase. The first experiments indicating that some plants do not use the established C3 cycle were done by Kotschak and Karpilov. The pathway was finally discovered by Hatch and Slack in 1966, so it came to be known as Hatch and Slack pathway. C4 cycle operates in the following steps. The first step is the conversion of pyruvate to phosphoenol pyruvate by the enzyme pyruvate orthophosphate dikinase. This reaction requires inorganic phosphate and ATP plus pyruvate. producing phosphoenol pyruvate AMP and inorganic pyrophosphate. So the enzyme involved here is different. It is not Rubisco, but it is PEP carboxylase. Oh, sorry, dikinase. To avoid photorespiration and excess of water loss, tropical plants operate an alternative form of photosynthetic pathway. Instead of Calvin cycle, which is the dark phase of the C3 cycle, C3 plants, a different pathway is followed in the C4 plants. It utilizes an enzyme different, which is not Rubisco. The first experiments indicating that some plants do not use the established C3 cycle. They were done by Hugo P. Korshak and Yuri Karpilov. The pathway was finally discovered by M.D. Hatch and C.R. Slack in 1966. So it came to be known as Hatch and Slack's pathway. C4 cycle operates in the following steps. The first step in the pathway is the conversion of pyruvate to phosphoenol pyruvate by adding phosphate to pyruvate. The enzyme involved is pyruvate orthophosphate dikinase. The reaction requires an inorganic phosphate and ATP plus pyruvate producing phosphoenol pyruvate, AMP and inorganic pyrophosphate. The second step in the fixation is the fixation of carbon dioxide into oxaloacetate by enzyme PEP carboxylase. So you see, instead of Rubisco, the enzyme used for carbon dioxide fixation is PEP carboxylase. The oxaloacetate is converted to malate, a simple organic compound which is transported to the bundle sheath cells. Inside the bundle sheath cells, malate is decarboxylated. Carbon dioxide, which was captured by PEP carboxylase, has now released it in the bundle sheath cells to produce carbon dioxide and pyruvate. The carbon dioxide now enters the Calvin cycle, which is running in the bundle sheath cells, and the pyruvate is transported back to the mesophyll cell. In C4 cycle, every carbon dioxide molecule has to be fixed twice. First by a 3-carbon organic acid, which is the pyruvic acid, and second by Rubisco. The C4 pathway uses more energy than the C3 pathway. The C3 pathway requires 18 molecules of ATP for the synthesis of 1 molecule of glucose, whereas the C4 pathway requires 30 molecules of ATP. In this figure, C3 plants and C4 plants are shown. In the C3 plants, both types of cells, both the cells are mesophyll cells, and in these you will see carbon dioxide is being fixed by Rubisco by the Calvin cycle or the PCR cycle. In C4 plants, two different types of cells have been shown, the mesophyll cell and the bundle sheath cell. In the mesophyll cell, the enzyme operative is PEPC, which means PEP carboxylase, phosphoenol pyruvate carboxylase, which initially takes up the carbon dioxide molecule to form malate, and this is transported to the bundle sheath cell where the carbon dioxide is released and it is fixed by the Calvin cycle, and the enzyme operative in the bundle sheath cell is the Rubisco. So carbon dioxide in the C4 plants, a carbon dioxide pump is operative. A CO2 pump takes carbon dioxide from the mesophyll and transfers it into the bundle sheath, which contains Rubisco and the enzyme of the Benson-Calvin cycle. C4 plants raise the concentration of carbon dioxide in the bundle sheath. The process raises the concentration of carbon dioxide in the bundle sheaths sufficient to saturate Rubisco with carbon dioxide and to eliminate photorespiration. Like all pumps, the C4 cycle requires an input of energy in the form of ATP. Recently, two terrestrial plants have been shown to have a single-celled C4 photosynthesis. This slide is a schematic representation of Hatch and Slack pathway, the C4 cycle. Two types of cells have been shown. The upper cell is the mesophyll cell and the lower cell is the bundle sheath cell. In the upper cell, phosphoenol pyruvate takes up carbon dioxide and forms malate. The enzyme is PEP carboxylase. Malate gets transported to the bundle sheath cell wherein it gives away its carbon dioxide, that is, it is decarboxylated, and pyruvate goes back to the mesophyll cell. Carbon dioxide which has been released by malate is now taken up by Rubisco enzyme, is catalyzed to form to run the Calvin cycle. C4 plants have a distinctive leaf anatomy. C4 plants have chlorophyll containing mesophyll and chlorophyll containing bundle sheath cells. This distinctive leaf anatomy of the C4 cycle, C4 plants, is known as the Kranz anatomy. The word Kranz means wreath or ring. Kranz anatomy is a specialized structure in C4 plants where the mesophyll cells are clustered around the bundle sheath cells in a ring-like fashion. The number of chloroplasts in the bundle sheath cells is more than the mesophyll cell. This is a comparative view of C3 and C4 plant anatomy. As you can see in the C4 leaf, there are distinctive bundle sheath cells showing, which is known as the Kranz anatomy. These are the differences between C3 and C4 plants. In C3 plants, the first stable intermediate product is a 3-carbonic acid, which is 3-phosphoglycerate or 3-phosphoglyceric acid. In C4 plants, the first stable intermediate product is a 4-carbon acid, oxaloacetate, which is then reduced to malate. In C3 plants, photosynthetic functions occur only in the mesophyll cells, whereas in the C4 plants, photosynthetic functions occur both in mesophyll and the bundle sheath cells. In C3, they require cool and wet environments. C4 requires tropical and dry environment. 95% of the green plants are C3 plants. Examples are rice, wheat, oats, etc. 5% of the green plants are C4 plants. Leaves of C3 plants do not show Kranz anatomy, whereas leaves of C4 plants show Kranz anatomy. C3 plants are common in temperate climates, C4 plants are common in tropical climates. C3 plants exhibit only granule type of chloroplasts, whereas a C4 plant exhibits both granule as well as agranule type of chloroplasts. Agranule chloroplasts are present in the bundle sheath cells. Some more differences are, in C3 plants, carbon dioxide fixation occurs only once, which is in the mesophyll cells, whereas in C4 plants, carbon dioxide fixation occurs twice. All the steps in C3 cycle dark reactions take place in mesophyll cells. The initial steps are carried out in mesophyll cells in C4 plants, and the subsequent steps are carried out in the bundle sheath cells. The optimum temperature for photosynthesis is comparatively lower than that of C4 plants. The optimum temperature for photosynthesis is comparatively higher than C3 plants. C3 photosynthesis is the oldest and the most common. C4 photosynthesis is a recent phenomenon emerging after the C3 photosynthesis. Photorespiration is not suppressed in C3 plants. Photorespiration is suppressed in C4 plants. Carbon dioxide fixation is slow. Carbon dioxide fixation is faster in C4 plants. Photosynthesis occurs when stomata are open. In C4 plants, photosynthesis occurs even when stomata are closed. There are also a few similarities between the C3 and C4 plants. Both the plants fix energy from sunlight. Both synthesize carbohydrates. They are the type of dark reactions of photosynthesis. The site of photosynthesis is the chloroplast in both the types of plants. This figure shows two different chloroplasts. The upper figure is of a granule chloroplast. The chloroplast shows grana stacks within the chloroplast, and the lower one has no grana within it. The agranule chloroplasts are present in the bundle sheath cells of C4 plants, whereas the granule chloroplasts are present in the mesophyll cells of